Hello, everyone, and welcome to today's webinar, Unlocking Ocean Secrets Marine Mammal Monitoring through eDNA. My name is Ashleigh, and I'm delighted to be your host for this session, brought to you by the Marine Mammal Special Interest Group, or the Marine Mammal SIG, at the IMarEST, the Institute of Marine Engineering, Science and Technology. Today, we're diving into one of the most exciting frontiers in marine science, environmental DNA, or eDNA, and how this revolutionary tool is transforming how we detect and monitor marine mammals, offering a non-invasive, highly sensitive method for assessing species presence across vast and remote ocean environments. Following strong interest generated during our recent Defra mNCEA Marine Mammal Monitoring Innovation Stakeholder Workshop, we're thrilled to bring you this dedicated session on eDNA and monitoring of marine mammals. Before we begin, I'd just like to give a quick reminder that we encourage you to submit questions throughout the session using the Slido feature, which is #marine, and there will be a link to that there as well. Please post your questions throughout the talk, and we'll have a dedicated question and answer section right at the end. And we'll address as many as we can. We've got fascinating speakers today, so I'm just going to share my screen and get this presentation live. There we go. Fantastic. So today's webinar, Unlocking Ocean Secrets, Marine Mammal Monitoring through eDNA. At the IMarEST, so there are certain strategic themes that the IMarEST follows, and there are many volunteers and many special interest groups that you can get involved with. But it is the largest marine organisation of its kind with over 12,000 members, and they are marine engineers, scientists and technologists. So bringing a holistic view on how we tackle certain aspects in the maritime industry, conservation industry. Generally, it's a really great collaborative organisation. Within that is the Marine Mammal SIG, or the mMCEA. This was founded in 2020. There are around about 350 corresponding members. 17 of those are very active members. And the group generally is to promote knowledge exchange, encourage collaborative partnerships, capacity building, and there are many different items that the SIG has worked on. They've done science communication articles, we've collated a career matrix, webinars such as this one, workshop, university guest lectures. We have the UN Decade of Ocean Science and Sustainability contributions, of which this event today is endorsed. And we have partnered with Defra, who are a UK statutory government body, with the Marine Mammal Monitoring and Innovation Project. And here we have the careers matrix. This was designed with marine mammal careers in mind. So we have set careers that could be within the industry, so within oil and gas, renewables. We do have conservation sectors, NGOs, sections, and also at the bottom of this marine mammal, the careers matrix, we have a list of actual profiles of people that are working in those careers now, and where they list their skills and qualifications to date to get to those careers. So do check that out, and there's a QR code for that there. Within the IMarEST, there are also many awards, bursaries and funding that are granted. Please have a look at these. We have the David Henson Inspiring Journey, Bill Loth Ocean Decade, student bursaries, particularly we have the IMarEST and MMOA, or Marine Mammal Observer Association bursary for aspiring marine mammal observers. So this is awarded on an annual basis, and we take on applications all throughout the year. We recently awarded that to the winner. So it'd be great to see more applications for this. And these funding and bursaries are available to you. So it's good to go and check these out if you are an IMarEST member. There are also many volunteering opportunities. So I'm a volunteer at the Marine Mammal SIG. You can join any of the SIGs as an IMarEST member. You can contribute to technical committees, consultations, or publications, support STEM outreach. I'm also the chair of the See Your Future group, or SYF group, and we're all about capacity building, outreach, and basically telling people about all these amazing opportunities and careers that are available and how this is potentially going to change with climate change. And, there are many different roles that are being navigated and changing in this new sort of sphere, particularly with AI as well. And yeah, it's just a great way to build skills and networks. Being a chair or co-chair really gives leadership and public speaking skills as well. So just something to have a look at as an IMarEST member, how to get more involved. And if you do have opinions on certain things, this is one great avenue for that. And if you are a student as well, so you do get free membership with the IMarEST. So do please scan that QR code or have a look at the membership link there. You will get that membership free and you get all these added benefits as well to do so. Okay, fantastic. So we have a fantastic array of speakers for you today. We've got Zora Sletsky, marine ecologist and eDNA specialist, Matt LeMay, research scientist at Hakai Institute, Matt Dempsey, business development manager at Dartmouth Ocean Technologies, or DOT for short, Nick Jeffrey, aquatic scientist at the Department of Fisheries and Oceans Canada, and Dr. Samantha Howlett, business development manager at Applied Genomics. So really fantastic array of speakers that we have today. We're going to start with Dora. Dora unfortunately couldn't join us today, but she has sent over her video presentation. So we'll go through Dora's now. And if you have any questions on hers, please use that Slido and we'll answer all those at the end. So I'll go to Dora's slides now. Thank you.
Hey everyone. Hey everyone. My name is Dora, and I'm going to talk to you about just a little bit of personal introduction. I am a marine ecologist and eDNA specialist at DHI Denmark at the Environmental Solutions Department. And as my background, I have a PhD in cetacean eDNA applications from the University of Copenhagen. All right, let's dig in. I'm going to introduce you to eDNA in the context of marine mammals through some of my research experiences and also some important studies that I will briefly mention. And I'm also going to talk a little bit about what our team is currently working on in terms of eDNA. Start with the very basics. What is environmental DNA? Environmental DNA, or as we will call it, eDNA from now on, is the genetic material of different organisms that can be detected in the environment, such as seawater, freshwater, sediment, soil, ice, or even air. Well, during this talk, we will focus more on the marine environment. So more specifically, when we take a water sample, the genetic material that we find in that water sample can be categorized into two groups. It is called organismal and extraorganismal eDNA. The organismal DNA is practically the creatures that we sample together with a scoop of seawater, for example, zooplankton and phytoplankton, that end up in our samples, and we will have their DNA in our extractions. Extraorganismal DNA is some cellular material or potentially free-floating DNA shedded by bigger organisms that also live in the sampled area. I know the title says marine mammals, but I am going to focus on cetaceans a bit more, since this is the group that I studied the most. So currently there are 9 species, 25% of those are threatened, which means they are either critically endangered or vulnerable. There are also massive data gaps of cetaceans worldwide, and that is due to several reasons. And just to name a few, it is due to logistical and resource challenges and the fact that surveys are also highly weather dependent. So we are definitely in need of some new methodologies that can help us out with gathering very important data of cetacean populations. I found a laser pointer, which might be helpful. So here you can see a figure that basically depicts the pipeline of eDNA sampling from fields to data. We go out with the boat, we take some water samples, and we filter the water samples to concentrate the genetic material or cellular material that we encounter in the sea. And then it's either filtered directly on the field, in the field, or we take the samples on shore and filter it in a laboratory. Then the DNA will be extracted from these filters and then amplified. And after the amplification step, it's possible to use these DNA copies to quantify the concentration of DNA from certain species or one target species, or these amplified DNA can be sequenced for identification of multiple species that are potentially present in the samples, and can also be used for population level inferences, such as population genetics. Now let's have a look at the different approaches to study cetaceans with eDNA. It's possible to target one single species in our eDNA analysis. This means that once the eDNA is extracted from the samples, it can be amplified in a way that it only amplifies the DNA of our target species, and then this either can be quantified or sequenced for species confirmation. This approach is often used for species-specific presence-absence studies of endangered species that are often very hard to detect visually or acoustically. In this example study here on the slide, researchers were targeting finless porpoises in Japan in a busy strait, and they were able to identify where the animals were most likely to be found based on eDNA signals. DNA has this inherent capacity that allows for sampling for the signal of multiple species at the same time. The method that harvests this opportunity is called metabarcoding and is commonly used as an approach to identify multiple species of a certain taxonomy group at the same time. For example, in this study that you can see on the slide, traditional and eDNA-based monitoring methods were compared and showed how they performed for marine megafauna. What you can notice here is that both methods show unique and overlapping detections, which means that it is an excellent complementary method to actually use both traditional and eDNA surveys at the same time. Population level inferences in terms of eDNA are somewhat challenging, and while this application of eDNA is not widely used because the endogenous, so the species-specific DNA content in water samples is quite low, and it's sometimes challenging to gather enough genetically informative DNA to actually use it for population genetics. But in this study that we did on bowhead whales in West Greenland, it was possible to obtain enough DNA from the close vicinity of animals to understand the genetic diversity of local populations in comparison to what has been done with long-term biopsy sampling and tissue sampling. DNA comes with certain bag of challenges that we have to consider whenever we are designing our surveys or when we're interpreting our data. There are several things that could affect the presence of eDNA in seawater, and some of these are physiological, so coming from an individual or species-specific level, or environmental, which are either physiological are promoting eDNA shedding, so eDNA production, and the environmental factors are contributing to the decay and eDNA degradation at the same time. And eDNA persistence in seawater is a challenge and opportunity at the same time. At least that's how I like to see it. It is a challenge because we might not be able to detect the species that are present in the area that we're sampling because of degradation. but it's also an opportunity because due to the decay, eDNA provides a relatively real-time snapshot of the sampled ecosystem. Now let me talk briefly about our work at DHI in terms of eDNA. Our team is currently working on integrating eDNA data of different marine communities into a biodiversity framework called the Biodiversity State Indicator. And the metric relies on classic biodiversity indices integrated with functionality and also all kinds of different interactions of participants of the community. Behind these indices, there is a curated trait database that supports the BSI function, such as the safest database of the North Sea seabed and fauna that you can see here on the slide, but we are working on a more elaborate curated database that can support the BSI score. And the BSI calculation provides us with a standardized score that translates to a % difference from an ideal or reference state, either in a positive or negative direction. Then this BSI score then can be integrated into reporting, perhaps connectivity models as well, and also all kinds of other ecological models. Thanks a lot for listening, and feel free to reach out if you have any questions, or check out the website of DHI if you would like to get more information about the company. And have a good rest of the day. Bye.
Fantastic. Thank you very much, Dora, for sending in your video presentation. Yeah, really, really interesting to see how we're having to look back to figure out how we can move forward with conservation of these animals. Fascinating. So our next speaker today, we have Matt. Oh, there we go. Matthew LeMay from Hakai Institute, and we also have Luz Rodriguez. Luz is just going to give a brief insight into the OBON network. And then Matt, if you want to take the stage, that'd be great. Although I can't see Luz there. Ah, there we go. Fantastic. Thank you very much, both. Oh, you're on mute, Luz. Sorry. There we go. Thank you.
Okay, sorry about that. Thank you, everyone. I'm Luz Rodriguez, and I'm the Ocean Biomolecular Observation Network Program Manager, which is a UN Ocean Decade program endorsing other projects. So this is a brief, just wanted to walk you through what what OBON is about. So we, in a nutshell, OBON's vision is to accelerate decision making to restore our oceans using the signatures of life on Earth, as you just saw from the presentation, which are the biomolecules. And we are also aiming to create a hub for biomolecular measurements of marine life. We are guided by four objectives, which are to build a coastal to open ocean biodiversity observing system using multi-omics and autonomous tools, to grow capacity through training programs aided by agencies and philanthropy, and third, to strengthen marine omics data systems and improve ecosystems models by adding biomolecular insight. A fourth one is to partner with conservation experts like you, policymakers, and the private sectors, so this work directly supports sustainable development. At the moment, we're gathering information and sampling locations where you will be able to find on our website. I'll share the links later. And we are currently mostly registering coastal observations, but in each of the UN decade calls, we encourage the endorsement of open ocean projects, and we are open to collaborate with one of research cruises that might not be sampling regularly, but now that they can be endorsed as contributions, as this webinar is, we are happy to endorse them on their own. We have a program coordinator, which is based in POGO, which is the Partnership of Observations for the Global Ocean. And we receive guidance from a SAC, an international scientific advisory committee, and an executive committee, both chaired by Professor Margaret Linen. And we are in the process of setting up a user advisory committee to ensure participation from different sectors towards our goals of accelerating the use of biomolecular science and technology. As I mentioned before, you can contact us either directly, I will share the links with you, or follow us on social media, including Twitter, Blue Sky, and LinkedIn. We also have our YouTube channel where you can find presentations, webinars like these from all the 21 projects that we have endorsed up to this moment. So thank you very much, Ashleigh, for the space, and I hand it over to Matt LeMay, who is also part of one of the projects that Aubon has endorsed.
Great, thanks. And hi, everyone. Yeah, I'm excited. Today I'm going to talk a little bit about some work we've done using genetic tools to understand exactly what whales are eating. Next slide, please. So whales have a huge, oh, go back, please. Thanks. Whales have a huge impact on coastal food webs, consuming several tons of their prey as marine invertebrates or fish per day. Next slide, please. And so there's really great interest in understanding exactly what it is that whales are eating. And there's a lot of conventional tools that have been used for this, specifically cameras mounted either on drones, underwater, or sometimes even attached to whales themselves. We can also look at stomach contents to infer what whales have been eating. And then there's also more indirect ways using sonar and different ways of trying to understand what's in the water and what's associated with the whales. Next slide, please. What I'm going to talk about today is some approaches where we've used genetics to try to understand whale diets. And so the theory here is the same as what Dora presented and what other speakers are doing, except that instead of starting with an environmental sample from the ocean or soil, we're actually starting with a fecal sample. And so you can see the picture on the left, there's the ocean. That orange blob is humpback whale poop, which we can collect. And then by bulk sequencing all the DNA that's in that fecal sample, we can then match those sequences to a database and try to understand exactly what all the different prey species are in that sample. Next slide, please. And so this is an approach that we've used on a number of different species and with a couple of really great collaborators. The work I'm going to present today is based on humpback whale study that was carried out with Rhonda Reddy, who was a PhD student at the University of Victoria. But we also have ongoing collaborations with Sarah Fortune from Dalhousie University, applying this approach to bowhead whales and also North Atlantic right whales. Next slide, please. And so as I said, today I'm just going to talk a little bit about the work we did with humpbacks. This was a project where we opportunistically collected fecal samples at different locations around Vancouver Island in British Columbia. These were collected by Rhonda herself as part of her PhD research, and also by whale watching companies who were out there to opportunistically scoop up fecal samples whenever possible. Next slide. And so the basic method for this is any time that a whale was observed pooping, the boat would go up when it was safe to do so, collect the sample, and then that sample itself was split into two. So on one half of the sample could be used conventionally for microscopy to look at any fish bones or marine invertebrates that were in there to try to figure out what they're eating. And then the other half was used for genetic sequencing to then use that as a way of inferring the diets of these species. Next slide. Thank you. And so just to show some results, this is from the microscopy side of the sample. And so the point of this is just to show that there's quite a large diversity of prey items within humpback whale fecal samples. I won't go into huge detail here, but there's little pieces of digested marine invertebrates. There's lots of different fish bones from numbers of different species. There's also some parasites that were detected and non-prey items like plastic and feathers and other things in the water. Thank you. And so just to contrast that with the genetic data, I'm going to show first fish prey, and then the next slide will be the marine invertebrates. And so the way that we interpret this figure is it's essentially a fancy bar graph where each bar in this figure is a different whale fecal sample, and the colors represent the different species of fish that were present in that sample. And so the take home from this slide, the main thing I want you to get from it is just that there is a great diversity of fish in humpback whale poo. And it's quite variable, suggesting that these are quite generalist predators when it comes to eating fish. Certainly Pacific herring is common. It's that sort of gray-blue color. But really, they're quite general feeders when it comes to fish. Next slide. Whereas these are the marine invertebrate species that were detected in the humpback whale poop. And in this case, it's sort of the opposite pattern, where overwhelmingly you can see that krill, which is that pink color, is a really, really dominant marine invertebrate prey item, no matter where the sample was collected. Next slide. And so just to sort of contrast the microscopy approach with the DNA approach, just looking at fish here, it was really kind of comforting and reassuring to know that Pacific herring and walleye pollock were two species that were the most abundant bones present and identified through microscopy, but also the most abundant DNA present using the genetic approaches as well. So both of these species were overwhelmingly detected across samples. Microscopy did detect the presence of northern anchovy, which was absent from our genetic data. Conversely, the DNA detected an additional 29 species that microscopy didn't detect. Most of these were very rare or only detected in one sample. Next slide. And then I just want to highlight a really great case study where we dug deeper into one of these whale individuals to further validate this genetic approach for studying what whales are eating. Next slide, please. And so for this whale, Rhonda and her colleagues attached a tracking apparatus to the whale so they could monitor it as it was doing its dives and as it was feeding. And so the way you can visualize this figure is it's essentially the whale's track through the water. At the top, you can see it at the surface, taking several breaths and then doing a deep dive where it's going up and down through schools of prey, fish in this case. And next slide. And so then overlaying the sort of purple line here that you can see going up and down is the whale's path going from the surface and then down to the bottom. And it's overlaid with the sonar where you can see in red the schools of fish that it's preying on. And so using these sonar approaches, they're able to infer that these were most likely Alaska pollock schools that were being predated on by the humpback whale. Next slide, please. Which matched really nicely to both the genetic evidence from this individual and the bones. So I believe all of the bones present in the fecal sample were from walleye pollock, and also 99% of all the DNA in the poop sample was also Alaska pollock. So it was a really nice case study where we could validate each of these approaches that gave us the right or the same answer. Next slide, please. So I'll just end it here, and I just want to conclude by saying that I think that these DNA metabarcoding approaches are a really nice way to study the diversity of whale diets. Some of the main advantages over previous methods are, first, that it's morphology independent. And so this means that we don't need to be able to identify a prey item to know what it is. So when we're looking under the microscope, the prey items can be really degraded or partially digested. And it's hard to put a species name on some of those. Whereas as long as there's a genetic sequence in there, we can match it to the database and know exactly what species it is. It's also really scalable. So if you're doing microscopy, 10 samples takes 10 times as long as one sample. But when you're doing genetics, you can pool many samples together into a run. And so it's quite scalable because you can run multiple samples in parallel. It's got good accuracy, and this has been validated by a few independent studies. And it's also, the field work is quite simple. It just requires a bucket to scoop up a sample. And so I think it's a really nice approach that people should consider when studying food web ecology of whales. Next slide. And I will end it there and pass it off to the next speakers. Thank you very much.
That's fantastic. Thank you, Matthew. Yeah, great talk. Just thinking about that validation technique as well. And I know I've heard about the great whale conveyor belt, bringing all those nutrients. And you could see that clearly in that humpback whale's track at the surface and then going down and foraging. Yeah, fantastic. Thank you very much. I can see the questions are coming in thick and fast on the Slido, so thank you. Keep them coming. You can, if you've got the same question as somebody, if you press like, it kind of upvotes the question in there as well. So we've definitely got plenty of questions coming at the end. Okay, next up we have Nick and Matt. So they're doing a sort of dual presentation. First we have Matt. So Matt Dempsey, the Business Development Manager at Dartmouth Ocean Technologies. And then we'll have Dr. Nick Jeffrey, Aquatic Scientist at the Department of Fisheries and Oceans, Canada. So Matt, I believe it's you first. Perfect, thank you. I'll let you take it away.
Am I coming in? Thank you so much, Ashleigh. Matt, that was an excellent presentation. I loved how you were able to kind of hammer home that eDNA, how powerful it is, but how it also can be used in concert with more traditional marine mammal monitoring techniques like acoustics and visual inspection to kind of come together and create such a comprehensive analysis over these marine mammals and what they're doing. Ashleigh, thank you so much. My name is Matt Dempsey. I'm the Business Development Manager, Technical Sales Manager here at Dartmouth Ocean Technologies. Don't worry, I'm not giving you a sales pitch today. I had to fight my baser instincts and come up with something that's a little bit more general for those of you who aren't very familiar with eDNA and sampling techniques and the ins and outs of it, and of course, auto samplers as well. So Dora did a fantastic job at explaining what eDNA is. I don't think I can improve that at all. But when I think about eDNA, it's typically traces that organisms leave behind in their environments. Like you and I are leaving eDNA in our environment right now with skin cells and hair. So you apply that to the ocean with marine mammals, with bacteria, and that's what eDNA is, and it can be found anywhere. There's a great quote by a researcher that basically said that the ocean is just a soup of genetic material from all the different organisms that exist in there. So think about that the next time you're going swimming in the ocean. And I'll get into during this presentation on how it's collected and touch on a little bit on how it may be used. eDNA filters. You can't collect eDNA without running the water, in this instance, that it's contained within over filter membranes. There are many different types. Some researchers prefer some over others. You've got from, I guess, top to bottom on the right side of the slide there, you've got encapsulated disc filters. Below that is a very common filter that's used called a Sterivex filter. And there are new filters being developed all the time, like this one at the bottom here by Cephalium. Much larger, much more water can be passed through it before it gets clogged. Filter sizes are customizable, and it really depends on what you're looking for and what kind of pore size you're going to select in your filter. If you're looking for bacteria or like single cell organisms, perhaps you would choose something with a smaller, in this instance, 0.8 micron filter, or even smaller than that. If you're looking for larger particulates in the ocean, you might be able to get away with a larger pore size filter. It seems to be researcher dependent and what they're exactly looking for. And the water volumes that researchers are looking for to be able to pass water over their filters, it varies depending on the research and what they're looking for. But if the DNA is more abundant in the water column, then you need less volume to collect that. But if the DNA is less abundant, then you're going to need more volume of water. But we found that about 50 to 20 liters is common for filter sizes. So once you collect your eDNA sample, you need to preserve that sample, because the jury's still out on exactly how long DNA will last for in its environment. RNA is even less. It's hours or days. DNA, we've found that it lasts between 12, 48, in some cases 72 hours, which is highly dependent on the specific environment that it's found in. how close it is, if it's in the photic zone or not, the metocean information and whatnot. So in order to make sure that your sample is going to be good for testing long term, it needs to be preserved. And there are a lot of different chemical preservatives that are available. RNA later being one of them, RNA shield, DNA shield. Some folks prefer ethanol. And again, it's researcher specific on which ones that are preferred. Whether it's cold water, the sample's collected in cold water, warm water, it varies. But basically what these preservatives do is they chemically freeze the DNA and prevent it from degrading so that it can have a longer shelf life and get to the lab for testing. Cleaning is incredibly important when you're collecting eDNA, and it's probably one of the more time-consuming tasks that the researchers have to do while they're out collecting eDNA. If you're doing station-based sampling at different coordinates and you collect your sample from the first location, if you do not clean your setup by the time you get to the second location, and you do take a sample there is a good chance you're going to have cross-contamination from the first sample and the second sample. And if you're looking for truly discrete samples, then proper cleaning protocols are extremely important. Cleaning solutions that are typically used can vary. We use hydrochloric acid in our system, but other folks are using bleach, peroxide, and I'm sure there's many, many more as well. So you collect your eDNA. Why did you do that? Well, because you want to analyze it for, the types of things that Matt LeMay was showing you just in the last presentation. There are a few different ways of going about this lab analysis, but there's two main tracks. The first track is qPCR testing, and you guys might all remember this from that wonderful COVID period of our lives, where you would stick a swab in your nose, and then that would be put into a solution and tested. Well, when you were in line, when they were doing that using the qPCR machines, that fluid that you put your sample in is a primer based upon that COVID genome. And what happens with that is it'll bond, if you do have COVID or did have COVID, that would bond to the DNA of the COVID. And then a qPCR machine is going to heat that sample up. It's to about 90 degrees Celsius, and then cool it down to about 30 degrees Celsius. And every time it does a heating and cooling cycle, that amount of DNA doubles, and it doubles, and it doubles. And after you do that about 100 times or so, then you can actually see it optically, that COVID DNA, and you know that you have COVID, unfortunately. One of the good benefits of qPCR testing is you can actually get a relative abundance from that as well. If you divide the DNA magnitude by the number of cycles your qPCR machine has done, then you can infer how much DNA you had on your filter initially, and then you can use that to draw correlations between populations, proximity, and all that kind of good stuff. And the other kind of testing is the metabarcoding, and that basically you take your sample and it will apply the genomes that were from that sample to a library of different genomes that have been recorded, and then it will tell you basically what species or populations you have in that one sample. Traditional eDNA sampling is done with a syringe, and it's done manually. So water is brought into the syringe, and then it's expressed over the filter over and over again until, well, the researcher gets tired or they have enough water that's been through the filter. So there's some pros and cons to that. Pros, it's very it's inexpensive. You don't need that much equipment to go out and do that. It's portable. You don't need any power. But it is time consuming. You get lower volumes across the filter because it's all manual, and eventually the filter will become clogged and difficult to pass water over. Another way that's typically done is through a pump, like a peristaltic pump that you see in the slide there, and that will pull water through a filter until it starts to struggle. The filter gets too clogged. It's fast, it's inexpensive, but it is more complicated. You need to have a power source. It's more equipment to bring around, and it's both of these sampling methods are only really good at shallow depths. So enter auto samplers. These are still fairly new on the market. There's only a handful of companies that are making them. Some companies have actually been doing it for quite a long time. And Barry, for example, has had an eDNA sampler for decades now. But they're becoming more commercially available and more inexpensive and accessible for researchers. So auto samplers are powered, they're fully programmable, and they're depth tolerant. But there's a bunch of different kinds of them. There's a wide variety of samplers. There's no one size fits all yet, at least. Some are good at station-based sampling, others are good at long-term deployments at depth, kind of set it and forget it kind of situation. But they all do basically the same thing. They're going to draw water in, and they're going to pass the water across a filter and collect environmental DNA. One of the great things about autosamplers is that they're extremely versatile when it comes to deployment. So autosamplers can be deployed on autonomous surface vehicles, which you see on the top left there. They can be put onto bottom landers, ASVs or UUVs, like the sloping glider you see on the top right photo there. They can be deployed from buoys and overboarded like a CTD system. So basically deployed to a certain depth, different depth profiles, and then recovered. So because of that, you've got a platform agnostic sampler that can be used to collect eDNA in locations during times after events that would have typically been impossible. And this is Dartmouth Ocean Technologies eDNA sampler. This is what a typical eDNA sampler will look like, and a lot of the commonalities between them. So moving clockwise from 12 o'clock, you have the inlet filter. That's what the water gets drawn through the inlet filter, and by the pump that's within the housing of the sampler. On the far right there, we have the reagent housing, and our sampler includes several different reagents on board. I'll get into that in the next slide. You've got your pressure tolerant housing. They're all going to be pressure tolerant, so they can be deployed at depth and operate. Typically, pressure housing will include a pump. There'll be some valves inside that control the flow of the fluid and the flow of reagents, if it does have that. And then there'll be all sorts of hoses and electronics and PCBs and whatnot. Our sampler has an outlet filter, which the water expresses through after it's been passed over a filter. And then on the far left there, we have a 9-filter removable cassette. So you'll be able to take 9 samples before somebody needs to interact with the sampler, recover it, and replace that cassette, or just recover it all together. And then the concept of operations of our eDNA sampler. So the process flow starts with the researcher programming it. So you can program it based upon a time and date for all 9 samples. You can program it based upon a depth if you're going to be doing profiling. So each nine filters can be programmed for specific depth. Or you can use a third-party sensor to trigger samples. So think about if you had an acoustic sensor in the water and you're listening for North Atlantic right whales. If you get a positive hit on a right whale call, then you can trigger your sampler to take that sample. Once you have all 9 programs based upon time and date, let's say, and a specific volume for each filter, you will deploy that in one of the aforementioned deployment methods, and you will go home and you will forget about it until that last sample is taken. So what will happen is the sampler deployed in situ the first sample will trigger, and when that does, water will be passed in over the filter, out the outlet filter, and until you hit that volume. Once you have that volume, we run into our cleaning and preserving protocols. So you've got your DNA on your filter, the pump and the valve tree within the system will switch over. It will draw a DNA preservative out of a reagent bag and into a filter, where it will chemically preserve that sample. After that, we'll do a prime cycle. We'll get rid of that RNA later out of the workings of the system, the lines, the valves, the pumps, and we'll put that into a waste bag so it's not expressed into the environment. Once that's finished, we'll do a self-clean. So we have a 5% hydrochloric acid compound on board that will be pumped through the works of the system, and it's held for about 5 minutes to get rid of any chances of cross-contamination. Once that's finished, it gets ejected out the inlet and out the outlet after it's diluted with milliQ or lab water. So that kind of helps keep the inlet and the outlet filter clean of biofouling. Once that's finished, we'll do another prime, and you'll be ready to go for sample number 2. And that kind of is a rinse and repeat for samples 2 through 9. Once you've got all your 9 samples, you can get up off your couch and go and recover the sampler, take your samples off of the cassette, install another cassette, redeploy, and then do it all over again. And that's all for me. I hope that adds a little bit of context to what Nick is going to talk about now. Nick and Fisheries and Oceans Canada have been incredible partners to Dartmouth Ocean Technologies. We're a small business, and they were early adopters of our system. We can't thank them enough for the work that they do, the papers they write on our sampler, and all the things they're doing with the samples they collect. Thank you all very much.
Okay. Thanks, Matt. Thanks for the introduction, and thanks to Ashleigh as well. So, as Matt and others have noted, my name is Nick Jeffrey with the Department of Fisheries and Oceans in Canada, based at the Bedford Institute of Oceanography, which is in Dartmouth, Nova Scotia. And today I'm going to talk to you a little bit about our marine mammal monitoring through general biodiversity monitoring of Canadian marine protected areas using both eDNA metabarcoding and qPCR. So in Canada, we have a number of marine conservation areas that are administered by different federal agencies. Department of Fisheries is the one that leads our marine protected areas specifically, as well as marine refuges, which are closed under their closures under the Fisheries Act. They serve different purposes, but they all contribute towards our goal of 30% protected marine real estate by 2030 coming up. So we have in Canada currently 15 what I'll call capital MPA marine protected areas that are designated under the Oceans Act, as well as numerous marine refuges and other areas for conservation administered through Parks Canada and Environment Canada as well. On average, these conservation areas are quite large, about 3,500 square kilometers and ranging to over 300,000 square kilometers up in the high Arctic. And it can be very challenging to obtain ecological baseline data as well as monitoring data for areas of this size over time. Our MPAs are also quite deep. On average, again, greater than a kilometer deep, which is difficult to monitor, especially with net-based or grab-based sampling, which is typically limited to the continental shelf in Canada. So we need technologies that are scalable and efficient, but can also sample in really deep waters quite easily to help monitor these areas over time. Next slide. Marine mammals are a big part of our marine conservation plan, and I'm not a marine mammal expert per se. We do have several great teams of marine mammal researchers that are dedicated to marine mammals through other methods such as passive acoustic monitoring, satellite tagging, and marine mammal observation. And so, being part of the Marine Protected Areas team here, we work with those teams quite a lot. And it's really helpful to have their expertise available, whereas we focus on genetic techniques for this. And so in the bioregion that I work on, which is the Scotian Shelf and Bay of Fundy off Nova Scotia, there's numerous marine mammals that are present. North Atlantic right whales are seasonally present and highly endangered. We also get other endangered species, such as northern bottlenose whales, which have a resident population offshore of Nova Scotia in a marine protected area called the Gully, as well as blue whale foraging and transit areas, and numerous other baleen and toothed whales that are observed along the area. So these are typically monitored through a number of different methods, such as at sea and dedicated flights, passive acoustic monitoring, opportunistic sightings that go into a database, helicopter flights that are primarily for seals, looking at seal populations, satellite tagging, and of course, environmental DNA. Next slide. There we go. And just so you can have some reference for some of these spots, we have the Gully MPA, which is a large submarine canyon offshore Nova Scotia, where there's a resident population of northern bottlenose whales. They like that canyon habitat for feeding and with other beaked whales as well as baleen whales that inhabit the Gully. But these bottlenose whales really stay within the continental slope canyons for feeding and don't move particularly far as we know. St. Anne's Bank is a marine protected area off Cape Breton Island of Nova Scotia that serves as sort of a migratory area between the open Atlantic Ocean and into the Gulf of St. Lawrence. So typically right whales are moving in and out of this area seasonally, lots of other baleen whales, and often quite a lot of pilot whales in this area. And then on the south shore of Nova Scotia, or in deep water off southern Nova Scotia, is an upcoming area of interest that's currently undergoing consultation to become a protected area called the Fundy and Channel Browns Bank, which also serves as a migratory pathway between the Atlantic Ocean and the Gulf of Maine for baleen whales, and serves as an area of importance for blue whale foraging. Next slide. In the coastal zone, we also monitor and include marine mammals in our biodiversity monitoring. So in a coastal area called the Eastern Shore, there's a lot of seals that are monitored, as well as dolphins and porpoises. And same in the Musquash Estuary, which is a very small 7 square kilometer marine protected area in the Bay of Fundy. It's a mix of freshwater and estuary water and marine water, but there is what we think is a resident population of harbor porpoise as well, and occasionally other species like small baleen whales and dolphins are seen there too. Next. So eDNA has been mentioned a couple of times from the past few presentations. It's very standardizable and easy to collect and efficient for monitoring a broad scale of diversity, everything from microbes all the way up to marine mammals, as well as deep water species that we don't typically get to sample very often. We in Canada, our marine protected areas don't allow commercial bottom contact fishing. And so we're looking for non-invasive ways to sample for biodiversity that's easy to collect and cost effective, minimally invasive, that can also monitor across diverse taxonomic groups. Next. And so we've been applying eDNA since about 2018 for monitoring as well as baseline information gathering. So we do our eDNA sampling using both a rosette sampler when we're further offshore and doing CTD profiles and take bottom and surface water samples with our rosette. We also collect water from the coastal zone using small boats or just poles from the beach and so on. And we're using this technology and eDNA metabarcoding in particular in basically all of our conservation areas, trying to create time series over time to see if we can monitor change in communities over time, as well as the introduction or loss of new species, including cetaceans. And so just some of these results here show that we can differentiate surface versus benthic communities using eDNA pretty easily. As well as seasonal and spatial patterns, again, using eDNA metabarcoding, so we can tell fall versus spring and summer animal communities apart in these different areas. Next. In our biodiversity sampling, again, we're typically trying to get as much diversity as possible, so we target invertebrates, fish, and cetaceans, marine mammals in general as well. So we haven't done much work targeting specific species, but we do get whales and other marine mammals in our samples using generalized primers for vertebrates. So we're able to detect those harbor porpoise pretty reliably in Musquash and northern bottlenose whales in the Gully. We'd like to continue doing this using some of the autonomous sampling that Matt mentioned. And so putting something like an autonomous sampler out in the Gully, we could then leave it for up to a year or so, let it sample monthly or something like that, and then come back and get it, which is great because then we don't have to go out there every month to sample. It's hard and expensive to get to these offshore remote areas. We've even detected ringed and hooded seal DNA in the coastal zone of Nova Scotia, which those species are not typically present. We don't know if that's just DNA that flowed down from Newfoundland or somewhere further north of us, or if there are, in fact, the occasional stray individual that come down here. So warrants further investigation. We also want to just really monitor that resident population, northern bottlenose whales, hopefully year round with using eDNA. Next. And so the auto samplers, like Matt mentioned, we've worked with DOT through an Innovative Solutions Canada grant, which was a great partnership. I don't work for DOT, but we have used their technologies. We did some testing with these samplers out in the Bedford Basin, which is Halifax Harbor, right outside our institute. Comparing these samplers over a 9-week period in the fall to regular water filtering using a commercial vacuum pump. And this was published by Mallory Van Weingarten, who's also with Department of Fisheries, in a journal comparing species diversity as well as preservation of the autonomous sampler and regular sampling. And so we're hoping that if we could deploy autonomous samplers out at sea and then be out on our boat, we also have been testing various portable labs for extracting DNA quickly, as well as amplifying or sequencing that DNA while we're at sea, because some of these steams on our ship can be very long, like days or hours at least. And so it's nice to be able to be efficient, and we could try doing some lab work, especially if we want to know if there's an endangered species of whale present right at that moment. So we can get potentially near real-time detections with our portable lab, as well as recovering the autonomous samplers. Next. Oh, and that's it. So thank you, everyone. I'm happy to stay for any questions.
Perfect. Thank you very much for that, Nick. Great to see the sort of evolution of that project. And it's sort of made me think about how we use acoustic samplers now, and we deploy them and leave them there for as long as we can, and then bring them up for, you know, a bit of SD cards and batteries and things like that, which sounds like this might be where the sort of eDNA sampling and technology is going. Fantastic. Thank you very much. Some of you may have noticed the time. We are 3 minutes to finishing, but we're going to stay on as we've got a presentation from Dr. Samantha Howlett, and we do want to get to your questions. We have many, many of your questions, so thank you very much for that. So we will continue, and we'll probably finish maybe 10, 15 minutes later than planned, but we definitely want to make sure that we get to all those questions. So thank you very much. We now have Dr. Samantha Howlett, Business Development Manager at Applied Genomics. Thank you.
Hi, Ashleigh. Thank you for asking me to participate in the webinar today. I'm Samantha Howlett. I'm a Business Development Manager at Applied Genomics. Applied Genomics are a marine eDNA specialist company. They've spent over a decade trying to develop techniques to best quantify uncertainties when doing eDNA in the marine environment. So I'm going to share today a few lessons learned from a couple of studies that we've done. So I'm going to do a bit of a deep dive on how you collect your environmental sample. So that first but very crucial stage of your eDNA study, how best can you do your survey design to make sure that the data that you're getting at the end of your study is as representative as it can be. So why is this important? Well, unfortunately, the reality is oceans and coastal habitats, when you're looking at marine eDNA samples, they're highly dynamic. So water moves. So you can have variability in your tides and tidal ranges for each of your sites. There'll be variability in currents and strength of those onshore currents. And then on top of that, not only does your environment move, but your species move. So once that DNA has been released, there'll be variability on your DNA degradation rates. So how far that DNA particulate matter will move. And yeah, so the question is, with all of this variability, how best can we design our eDNA survey to make sure that we're accounting for all of that variability? And we do this quite well when we do freshwater eDNA. So when we think of lakes and ponds, we have a known volume of water, and we know how many samples we can take to make sure that's representative. When we have lakes and rivers, rivers and streams, when you have water moving in one direction, you can understand flow rates to understand how far eDNA will travel, and you can build your eDNA survey around that. When we think of quantifying uncertainty for eDNA in the marine environment, quite often the focus is on, when at your tidal cycle are you sampling? Can you keep that consistent to make sure that it's comparable? Maybe you focus on slack tide to try and make your eDNA signal as localized as it can be. And then finally, just collect more data and do more samples. Yes, the more effort you put in, the more you're going to cover that variability in the data. But quite often, as you collect more samples, you get more data, but the costs will scale as well. And so to get good data in the marine environment for eDNA at scale can become expensive quite quickly, but it doesn't have to. So what I'm going to do today is talk about two studies that compare different ways of collecting an eDNA sample. Why is the sample collection stage so important? Well, it's high risk and high expense. Quite often you've invested a lot of money to get people on site to collect that data. And if for some reason you don't get data from those samples, you can't go back. You definitely can't go back in time. And even to get back to site can be very, very expensive. So collecting that sample and making sure you get good data from that, there's a lot of pressure on that. And as I mentioned, you want to make sure that when you do collect that sample, the data that you have is representative of your site. And quite often what we see with eDNA is a cost versus effort versus data quality. The more effort you put in, the better data you'll get, but it can get more expensive. And it's really important to mention for the sample collection, once you have that sample, no amount of wizardry and alchemy in your pipeline will account for inadequate sampling. So that's why we say at Applied Genomics, you can't detect it if you don't collect it. So it is really important to make sure that that sample collection is representative. So I'm going to compare passive sampling with active sampling in the first study, and then I'm going to compare for active sampling, high volume versus small volume sampling. So for our passive versus active, this was a Defra-funded study for the Marine Natural Capital Ecosystem Assessment that we did with Natural England. When I'm talking about passive sampling, I'm talking about metaprobes. So these are a 3D printed ball with a gauze inside that you tow behind a boat. As you tow that filter, the act of towing pushes water through, and that's how you collect your eDNA sample. At the same time at those sites, we did active sampling. So as the name suggests, this is actively pumping the water through a filter. This is also can be called encapsulated. Unlike your metaprobes, once you've collected your sample, it's not exposed to the external environment. And so it controls for cross-contamination much more effectively. So to standardize the passive sampling, what we did was we towed the metaprobes behind a boat for 2 hours at 9 knots to try and keep it consistent. And then for the active, we collected samples using Niskin bottles, and we did 4.5 litres, and we did 3 replicates per site. The first thing to point out is that we got far more species with the passive sampling than we did with the active sampling. And this was continuous across all of our sites. So when we think of why that is, what we really need to think about is we've got a very different survey design here. The passive sampler is basically a transect sample. So you're going from one point to another, whereas your active is a point count. You're basically dropping a bucket and saying, what have I got here at this point in time? So you've got huge variability across your exposure time and spatial coverage. When we think of the volume that's processed of water for your active versus your passive, 13.5 litres per sample per site for the active, versus however many litres went through for 2 hours towing at 9 knots. It's going to be an order of magnitude higher. So the volume of water in the area that you cover is probably why the passive sampler detected more species. Also with the passive sampler, as Matt mentioned, cross-contamination can be a really big issue. And this is what we saw with the passive sampler. There's a number of steps in the sample collection and sample processing stage that you have to account for. Otherwise, you can really introduce a lot of cross-contamination. So what we found was, yes, we found more species with the passive, but it was very challenging to standardize, taking into account speed, that you can't really account for the current flow and direction, and then how to mitigate against cross-contamination. The question then became, can we take the active sampling approach? Can we improve on this encapsulated approach so that it can process more water and become more representative? And through a lot of work, this is what we came up with at Applied Genomics. So this is our in-depth sampler. It's a programmable unit that will sample over whatever period of time that you want, and you deploy it, and you come back and collect it. So why this is important is quite often we will program this to sample over a 24-hour period. By doing this, we extend the sampling window to increase detections, can cover nocturnal activity, but more importantly, it covers 2 tidal cycles. So the environment moves around our sampler, and we account for all that variability in the data. So what we created was something that's really easy to use. It's quite simple. It's very affordable, but it improves on the efficiency and economy of scale for monitoring in the marine environment. So then we wanted to compare what the data looked like for your small volume versus your high volume. So we did a study at Poole and Plymouth Harbour. We put 1 high volume in the middle of the bays, and we did 3 replicates around the bays for the small volume. We focused on different habitats within the bays. So rocky reef, seagrass, and sediment. We wanted to see if we could detect differences using the small volume for different habitat types within the bay. And what we found, surprise, surprise, the more water you process, the more species you detected. So we absolutely saw that with the high versus small volume sampling. And what we found was really an economy of scale. So not only was it cheaper to deploy the in-depth sampler, it's more than half the price of doing all of your replicates for the small volume, but we actually found a 7-to-1 efficiency. So when we looked at the amount of species detected for each of the approaches, we found that you'd have to use probably 7 of the small volume samples to get the same level of detail for your high volume pump. So much, much more efficient and effective. And just finally, when we statistically compared the communities for each of the habitat types, we couldn't distinguish between them. So what your eDNA signal is giving you is a biodiversity profile of the bay and the habitats that make up that bay. So when you interpret your data, you have to make sure that you're doing that at the right scale. So just to summarize, for your passive, it's a very cheap and simple design, but there was huge contamination risk. It was really challenging to standardize across all of your surveys. And so there was high contamination risk. So the encapsulated method is much better for that. Small volume is seen as the conventional approach, but it's very expensive when you start to scale because you've got to keep doing replicates. And it's challenging to make sure that you're getting a representative data because you're really only going out and getting a snapshot in time. So that single point. So it's very ineffective. So with the high volume sampler, it's simple, it's effective, it creates a representative data, and it's standardized. So the amount of water that the pump pushes through is always the same each time. So the effort is standardized. The cons of that you have to go and pick it back up the next day can be a challenge in certain situations, but then also we have clients that will deploy this for like 4 to 6 hours to process those 50 litres, and you're still capturing order of magnitude higher species. Your effort is much higher than replicating your small volume. Very, very finally, so we focused on the first part of your data journey. There's a lot of importance in the second half of your data journey, so how you report that data. First of all, make sure that the lab you're working with is using the most up-to-date bioinformatics. This is reporting amplicon sequence variants, not OTUs. There's a number of reasons for this that create uncertainties. You've got arbitrary thresholds, all sorts of things. Happy to discuss more, reach out to me. And finally, data transparency. So we should always be clear with our clients on the quality of the data that we provide them at the end. So one of the things that Applied Genomics have is we will give you credibility scores on each of those taxonomic assignments when we give our clients our data. And finally, we put caution into using relative abundance as a measure for actual abundance. Again, there's lots of steps that introduce uncertainty in that. We focus on haplotype diversity and within species sequence variants as a measure of effective population size, which is a much more efficient way, or that it's more designed for what eDNA is actually designed to do. But that's a bit of a whistle-stop tour. There's probably lots of questions that people have on that for all of the points that I've raised above. Please do feel free to reach out to me and discuss this in more detail. And yeah, thank you for your time, and I look forward to continuing the conversation.
Thank you very much, Samantha. Thank you for sharing about the practicality of actually collecting those samples. Really interesting to see how you've developed that high volume active method. So thank you very much to all today's speakers. I'm aware that we are running significantly over time. I should have allowed more time for this webinar today. If you're still with us, we're going to do the Q&A now. I'm aware that we are over time, so appreciate those that are able to stick with us. But again, this will be recorded, or it is being recorded now, and you will be able to watch the Q&A back. So I've got the Slido open in front of me, and I would like to invite all the speakers to put their cameras on and come back, please. That's fantastic. Thank you very much, everyone. So we've had, I can't even count how many questions on the Slido. We've had 30 questions throughout the whole presentation. So thank you very much for everybody sending those in. I've shared, Luz also shared her link for the OBON project as well. I've put that on there if people want to see. I'm just going to dive straight into the first question. Matt LeMay, I think this one's for you. Could the diversity of fish present in humpback whale poop in BC also be due to the remains present in the water or in the environment in which the sample was taken, and not simply because the individual consumed them? How do you determine the difference?
Yeah, that's a great question, and I probably should have mentioned that in my talk. In that study, the PhD student also collected a control sample, so essentially a seawater sample that we could then be subtractive with, so we can look at what was present in the fecal sample and essentially subtract what was in the seawater sample to help strengthen the inference of what's actually in the fecal sample itself. And so for those prey species that were most abundant and confirmed with other methods, there was huge numbers of DNA sequences in the fecal sample that were either absent from the background water or at much, much lower levels and not as concentrated as they were in the poop.
Fantastic. That explains it perfectly with the control sample. So yeah, thank you very much for the question. We've got a question from Liz. For the more elusive deep diving cetaceans, e.g. beaked whales, is DNA for eDNA obtained from strandings, or are there other sources? And I know we had mentioned from Nick about the northern bottlenose whales, so you may want to take it on the deep diving cetaceans. Or Nick
Sure,
or
yeah.
Matt.
I'm not sure if that means like the reference material for comparing eDNA to, which could have been from strandings, but there also are tissue sampling expeditions using like a little dart or something that people collect for looking at health of the animals as well. So there are tissue samples taken on occasion. If talking about the eDNA itself, like, yeah, these northern bottlenose whales dive quite deep. They feed on mesopelagic squid and other fish and things in these canyons. But we are able to get their eDNA from throughout the water column. They're often at the surface as well, obviously to breathe, as well as down into the mesopelagic. I don't know if we get their DNA all the way at the bottom. I'd have to look at some of our older results, and we've got some hopefully new samples coming in from the gully. But we do detect them sort of at least in the top couple hundred meters of water, and there is some diversity in them. So we would like to apply eDNA to studying population health as well and genetic diversity.
That's great. Thank you, Nick. We've then got a question from Aditi, but I think this was answered, Matt, with they say, how do you separate fecal DNA to the eDNA present in the water, which I think you've mentioned is through a control. We've got another question. Are there any studies planned using stable isotopes and eDNA combined? Matt, I think you mentioned that in your sort of inferences. But does anybody know of any other studies that are being looked at, or is anybody working on any collaborations in this space? I take silence as a no. That's absolutely fine.
Our group does a little bit of stable isotope work, but not necessarily related just to diet, more for, I guess the stable isotope work is looking at food web ecology and energy transfer through the ecosystem. So for us, we do eDNA sampling at the same time, but they're kind of like complementary, I guess, and we don't directly compare them to each other. They're just two tools that we use for ecosystem and community diversity monitoring.
Fantastic, Nick. I think that's a great way to sort of define the two, as the two tools that we're using to work out more about the environment. Samantha, I'm going to come in for you for this question, just because you mentioned about having this element in your study and refining your tool. Could eDNA be applied in high flow environments, e.g. tidal channels, by combining hydrodynamics and decay models to identify source areas?
Yeah, and we've actually had a number of chats where we would like to collaborate with DHI on this. So Dora mentioned their eRewind. So the challenges that you have is firstly understanding what your DNA footprint is when you're collecting your sample. So you have your point where you collected it, and then you've got your probability map of where it could be sourced from. So you have to look at the local hydrodynamics for that. Our way of sampling over a tidal cycle just means that you account for all of the variability of how the water moves much more efficiently than if you do it at one point in time. And it just allows you that when you do those hydrodynamic models, it's much more representative and it's much more effective. So yeah, I hope that answered your question.
No, that's great. Thank you very much. I'm going to put a cut-off time. I know this is everybody's time, so thank you so much for the speakers, but we're going to do one more minute, if that's okay with everyone, and we'll cut off at about 25 past. So I'm going to go in with another upvoted question, and I think this might be to Matt LeMay as well. Did the use of sonar disturb whale M, or did the animal ignore it in your study?
I don't know. I wasn't out on the water for that. I don't believe so. The sonar is, a surface mounted thing, and it was just the tracker that was attached to the whale. But I'm sorry, I'm not familiar with the methods.
No, don't worry. And I think through these conversations, I'm seeing how, quite a few people are talking about real-time monitoring, and I work in the sort of sphere of mitigation and real-time mitigation. And I'm seeing some way that this could actually be used as a tool for real-time mitigation as well, just based around Liz's question there. There's quite a few questions about sort of careers and how you get into these types of careers. So maybe that's one that I could put out to the speakers after the talk, if anybody has any insights on what the sort of skills that have led you to your positions now. But quite a few people saying thank you very much to all the speakers. Very interesting. Lots to think about and further reading required on this. So yeah, we'll end it there if that's okay. Oh, go on, Samantha, sorry.
No, I just wanted to check. So I'm not sure if the question was specifically relating to estuaries and tidal channels, but one of the ways that we've used our programmable samplers in highly tidal estuaries is to deploy two samplers, one sample as incoming tide and one sample on the outgoing tide. And that's a really efficient way for you to kind of standardize across those two tidal movements. So happy to talk about that in more detail if anyone else has any questions.
That's great. Thank you very much for clarifying. But yeah, we're going to finish the live stream now. But firstly, just a huge thank you to the speakers for agreeing to take out time to deliver this webinar today. Thank you to those of you that have registered. I know that we've got nearly 500 people that have registered for today's event. So thank you so much. And thank you to the IMarEST and the Marine Mammal SIG for hosting today. So yeah, thank you very much, everybody. Enjoy the rest of your day.
Thank you for all the hard work, Ashleigh.
Thanks, everyone.